In this lecture, let us look at some practical issues related to fMRI, the history of fMRI, and some current research topics. With functional MRI, we're generally trying to observe the brain function, and so there's a bit more complicated setups. So for example, we have a video screen where the person can, by looking up, be able to see things on the screen. We can have them do particular tasks from that. Now this screen could be either towards the foot of the bed, or it can be actually towards the head, going out the other way, and that's actually the arrangement that we have for most of our research scanners. In addition, the person could be wearing some headphones where they're getting auditory information or some auditory stimuli. And finally, it's very loud in the MRI scanner, so it's difficult to hear what a person might be saying. And in addition, if the person would be speaking, they tend to move their head a little bit. So we generally have a button response box where they can provide some sort of feedback. Now, with typical MRI, we would spend several minutes acquiring a nice high-resolution image, such as the T1 weighted image shown here on the left. However, with functional MRI, we're trying to look at the changes in the brain over time. That is, comparing those times when the person is performing a task to when they're not performing a task. So in order to track these changes over time, we collect the images much more quickly, sacrificing, in this case, some spatial resolution. So the images will look a little more blurry, but instead we can acquire an image, the whole volume of the brain, every second or two. And then we look for the changes in that signal over time. So in this example, we have the person tapping the fingers on the left and right hand at the same time. And in this case, in particular, they were resting for 20 seconds, then tapping their fingers for 20 seconds, resting for 20 seconds, and so on. And then we look for where does the signal change in this pattern, that is increasing when we have them perform the task and decreasing in the intervening time periods. And then we can indicate those areas with color. And as you can see here, the areas in the motor cortex showing this characteristic signal change matching our task, and areas outside of that not showing those signal changes. We can then plot these changes on top of a nice high resolution image that's been acquired separately. And we can even render that, have it rotating around. And you can see here the activation in the motor cortex. If you actually look closely, you'll also notice activation in the visual cortex. And that's because we are showing some cues, some flashing lights of when the person was supposed to be tapping their fingers. This is an example of the very first fMRI image that was ever created from Jack Beliveau, shown in the cover of Science magazine in 1991. Interestingly enough, this was actually acquired using a slightly different technique than what I had described. This particular image was acquired using contrast agents. That is, if you inject a bolus of a contrast agent, that bolus of contrast agent moves through and causes the signal decreases as it passes through the brain. So they first injected this contrast agent while the person was lying at rest in the scanner. And you can see this decrease in the MRI signal and then increase again. The area under this is directly proportional to the blood volume. Then they showed some flashing lights to the person, and you can see this curve as shown here, dipping lower down, showing a bigger decrease, indicating an increase in the blood volume. The blood vessels have dilated. In addition, you'll also notice that this curve seems to be shifted to the left, and that's because the blood flow velocity has increased. When they make a difference of these two blood volumes, you get the image that's shown here in C, and that's showing the activation, the increase in blood volume in the visual cortex when these flashing lights are taking place. Now, interestingly enough, only a short time after this was demonstrated, it already was obsolete because several groups have shown that you can get similar measures of brain function, but using just the endogenous contrast, that is the blood oxygenation, rather than having to inject a contrast agent. So the first three papers, all of which came out about the same time, one from Ken Kwong from the Massachusetts General Hospital, one from Peter Bannattini and colleagues from the Medical College of Wisconsin, and from Seiji Ogawa and colleagues from the University of Minnesota. Now, if you look at the details, I believe Ken Kwong, in discussions that I've had with Peter at least, Ken Kwong was perhaps the first to actually demonstrate fMRI, although Peter Bannattini and the group at the Medical College of Wisconsin were the first to do this with echoplanar imaging. This is a movie of the very first fMRI images that were created. It was shared from me from Peter Bannattini. And you can see the alternating tapping and moving the fingers on the left hand versus moving the fingers on the right hand. And the moving the fingers on the left hand activating the right motor cortex, and vice versa, the right hand activating the left motor cortex. I also want to draw your attention to a really interesting special issue of NeuroImage that came out about 8 years ago in 2012, just 20 years after the discovery of fMRI, entitled 20 Years of Functional MRI, the Science and the Stories. What's interesting in this particular special issue is that they asked several of the people who had made some contributions to the field of functional MRI to write review papers about their contributions. But interestingly enough, they should also share the stories. What was it like in the early days of fMRI as they were making these discoveries? And in particular, Peter Bannattini has a paper in this entitled Sewer Pipe, Wire, Epoxy, and Finger Tapping, the Start of fMRI at the Medical College of Wisconsin, where he goes in detail about all of the interesting early developments of fMRI right here in Wisconsin at the Medical College of Wisconsin. Now, this was actually, functional MRI was actually developed while he was a graduate student. Now, one of the challenges that they faced is that the MRI scanners at the time did not have gradients that were powerful enough to acquire echoplanar imaging data. So instead, Peter Bannattini, together with another graduate student at the time, Eric Wang, actually built a local gradient coil. And this is actually quite a feat because they not only had to build this local gradient coil, but also design it and write the computer programs to design this particular gradient coil. Then actually build it by hand. You can see images from that here. This is from that special issue. And then also program the echoplanar imaging pulse sequence to run. They did all of this in a remarkably short amount of time, and then were able to collect images. And these are actually some pictures from Peter Bannattini's lab notebook from that time that is also shown in that special issue, really highlighting a lot of interesting parts of these very early fMRI experiments. So I would highly encourage you to take a look at that special issue and look at the papers within that. Currently, the most common application of functional MRI is for presurgical mapping. That is, when a neurosurgeon wants to go in and operate on a particular region, let's say to excise a particular tumor, then they'd like to know what critical functions are there in the tissue immediately surrounding the tumor. And if there are particularly critical functions very close by, such as the ability to walk or move or have language capabilities, then the surgeon may be more careful in those particular areas and try to spare that tissue as much as possible with the risk of not getting rid of all of the tumor cells. What kind of research questions are currently being asked with fMRI? Well, for one, we could look at methodological improvements. That is, how can we improve the images? How can we extract more information? A lot of my research is along these lines. Next, we can try to understand more about the normal brain. How does the brain work? How does it change during development? And finally, we can look at what's different in various disorders. How does the brain change during disease progression? Or what other brain changes during treatment? And can we actually detect differences in those who respond to a particular treatment versus those who don't respond? So these are just some of the examples of the kinds of questions that we